Our last speaker is Professor Charles Baroud he's a professor of the mechanics department at École Polytechnique since 2001. He also leads a joint research group at the Institut Pasteur in Paris. His work focuses on using microfluidics and soft matter physics to address a wide range of problems in quantitative biology. And that includes understanding what determines the structure of cellular aggregates in 3 dimensions and the dynamics of microbial populations. He's also the co-founder and scientific advisor of 2 startup companies. And with that, I'm going to turn it over to him. Thank you, Charles.
So thank you very much for the invitation. It's really a pleasure to be here and to represent Europe at this webinar. Just before I start with my scientific talk, let me say a couple of words of where I work. One of the labs I work at is called l'ADIX, Laboratoire d'Hydrodynamique of École Polytechnique. And since I've joined l'ADIX, there has been a very long tradition of publishing in JFM. Since the 1990s, there has always been an editor of JFM from the lab and on subjects of classical, let's say, fluid mechanics. But since this logo in the 1980s until now, things have modernized, let's say, a little bit. So the logo has changed, but also in addition to the traditional subjects that people work on at l'ADIX, there's been quite a bit of new activity on kind of more recent fluid dynamics problems like energy, biofluids, fluid structure interactions like wind-plant interactions, and so on and so forth. And I think that we can really see a transition from classical JFM subjects to subjects that could find really their place in flow. And so I think that this transition could be very much accompanied by the new journal. So what I'm going to talk to you about today are subjects along these new lines that you are seeing. I'm going to talk to you about biofluids and microfluidics. And so what I hope to show you is a little bit how we were able to go from very much fundamental fluid dynamics, multi-phase flows, to really quantitative biology questions along the last few years. So since this is the last talk of the session, I hope it's going to be a lot of movies. So I hope you have some popcorn. You can just sit back, relax, enjoy the videos. When I think about microfluidics, one of the ways to think about it is to think that we're going to replace tubes that you can hold with your hand and pipettes that you can pipette with your hands, and with something that's already that's integrated. And this, of course, exists already in most biology labs with these multi-well plates. But now the question with microfluidics is, can you replace these plates that have microliter volumes and a few conditions, let's say 100 conditions, with plates that use nanoliter volumes or less with tens of thousands of conditions? And the reason why this is interesting is not only because we're reducing the volumes, but it's because we're integrating the operations and performing many of them. And in this way, the idea is to kind of ask a new type of questions, not only ask the same questions with lower volumes, but really ask a new type of questions that you wouldn't be able to answer with traditional methods. And this is now very old. You know, this is 20 years old now, these pictures. But since the beginning of microfluidics, these ideas were present, but they were not. It took a long time for them to mature. But then there's a second idea that emerged really early on in microfluidics, which is to say, well, rather than making complicated devices where we have moving parts and so on, can we instead use fluid mechanics? Can we use droplets to encapsulate whatever reagents we want to study, and by following a particular droplet, follow the reaction that we want to study? And so early on, many people in the field identified some key problems that would be solved by the droplets. But in particular, one that I particularly find interesting is the number 4 on this slide, which is that if you have a droplet, it gives you a natural way to move a very small volume, which is the volume that's encapsulated by the droplet. And so you should be able to use a surface tension that's separating the inside from the outside of the droplet in order to manipulate very small volumes that are encapsulated. And so if I go back to this picture of the multi-well plate, the question that we asked in my group was, can we replace this complicated plastic device with droplets of water and oil, where each one of the droplets would now be considered as a separate microreactor in which we perform different reactions. And so what I'm going to show you first is how we do this, the fluid dynamics basically of the problem. So we're going to work with confinement gradients, and this is, the nice thing about this is that the physics is very simple. You'll understand it immediately, but then you can start playing many games with this. So as I say, the physics is simple. If you consider that you make a droplet of water and oil, for example, then the volume of the droplet is fixed, but by changing the shape of the droplet, you can change the surface area surrounding it. And the surface area corresponds to an energy. So there's an energetic cost of changing the surface area of the droplet. So simply by changing the shape of the droplet, you can change how much energy is stored inside it on the surface. Now, imagine that you take a droplet and you place it in a device where it's more confined on one side and less confined on the other side. Well, then in that case, you get a force because you have a gradient of energy. And the force is just the gradient of the energy with a minus sign in front of it. So this is like a gravity field, basically. The weight of an object is the gradient of the gravity. Remember freshman physics. This is very, very classic fluid mechanics. What's a little bit less classic is that now we can make devices that are flat everywhere, that are very thin everywhere, but then locally they contain an indentation in which the droplet can release some of its confinement. And if we do this, oh, no, sorry. So, okay, I'm sorry, guys. My assistant is here. So now we can take a droplet and have it confined everywhere, except locally inside of the little groove that can reduce some of its confinement. So, apologies. So when you do this, if you do this, then the droplet feels a minimum of surface energy when it's above this groove. And so it's attracted to it because it has a minimum of energy. And if you do this now in a microchannel, you can draw different shapes of grooves. And so this is what happens. This is an image of a microchannel seen from above. What you see in this little gray zone is the groove that's like sinusoidally shaped. And what you see is a droplet that's going to move from the left to the right and is going to follow the groove because it's attracted to it by the surface energy. And so in this way, we can remove the side channels and we can use confinement gradients in the vertical direction of the microchannel to manipulate the droplets to move them around. And these we call rails. If now we make a very simple rail, which is just a little hole, then we can use this as a position that we can attract the droplet to again. And then in this way, we can make droplets, and without using any structures in the plane of the microchannel, we can hold them stationary because of this reduction of surface energy. And so we call these anchors. And so in this way, these are quite simple microfluidic devices to make, but in this way, we can remove the side channels and we can have manipulation of the droplets everywhere in the channels simply by using the topography. So as good fluid dynamicists, what we wanted to learn initially was how do these devices work. So I won't show you any of the technical details, but I will just show you that you're able to put one of these droplets in a device and you push it at different velocities. And as you increase the velocity, you see that the shape of the droplet changes. And if you move the velocity high enough, then you detach the droplet completely. And so we had fun writing a simple mathematical model about this. And what you can find, for those of you who remember capillary numbers, what you can see is that if you equate the drag force with the surface energy force, you get that there's a critical value of the capillary number where mu u over gamma reaches a critical value. And below this critical value of the capillary number, you can anchor the droplets. And above them, you do not anchor them. So I'm just showing you to impress you, to show you that we know how to do the math, but I won't go into any more of the details. What I'll try to show you now is that this is really useful because since you make these with microfabrication techniques, then you can make many of these little holes everywhere. And then if you bring many droplets in parallel, you can very, very easily make an array of droplets that will just stay there because they are anchored in place. And you can watch the contents of these droplets for very long times, as long as you want, really. And if now I push the droplets out, I can remove them and repeat the experiment. Okay, so this was motion. Can we actually make droplets in this way? Well, the short answer is yes, we can make droplets in this way by connecting a microchannel to a region where there's a confinement gradient. And the physics is a little bit subtle, so I won't go into it. But the bottom line is that if you make a channel that has even a 1 degree slope downstream of the microchannel, then by pushing the water phase into the oil phase, you can detach droplets in a very, very regular manner. And their size is extremely well controlled because their size is almost exclusively controlled by the shape of the microfluidic device. It doesn't matter very much how hard you push. And so in this way, you can generate water droplets in oil without pushing the external phase, by simply pushing the internal phase, and the systems are extremely stable. And because this is extremely stable, then you can parallelize these channels into a single device. And so you can make a high throughput production of droplets. And I'm just going to show you a little application of this quickly, where you make all of these droplets that contain some magic mix, basically, so some soup that allows you to amplify. Oh, everybody knows what PCR is now because of the COVID. But so if you include the PCR mix inside of these droplets with whatever sample you want to analyze, you can make the droplets on a chip, you can run the PCR, and then you make a picture like this, where you measure the presence or not of the DNA that you're looking for in the droplets, and you can get an extremely precise quantification of the DNA that you had in your sample. So this is really a technology that works. We set up a startup company that sells this. And the point here is that these are technologies that emerged from a very fundamental study of fluid dynamics, but that actually have applications to health and other biological applications. So in my title, I promised to talk to you about immune cell dynamics. And so what I would like to show you now is much more recent work that we've been working on with cells on how to actually understand complicated collective behaviors of cells in these devices. So the way that we do this are devices that are a little bit different from what I showed previously, where there's a drop production region, some rails to distribute the droplets, and then a region for trapping the droplets and observing them downstream. So rather than explaining this in detail, I will show you another movie. What you're looking at now, on the bottom movie, what you're looking at is the third inlet from the right. So if you count 1, 2, 3, you're looking at the third inlet from the right. And what you're going to see is a real-time movie of how we produce droplets that contain cells. So here we go. At T0, I start the syringe pump, and here it goes. And what you're seeing are the droplets. So we generate droplets inside of these devices, and each one of the droplets contains about 100 cells. These are little white points that you observe. And now the droplets reach the rails that distribute them laterally in the device. And as they reach the rails, then they're just being pushed, and now they reach the region where they are trapped. And so each one of these droplets is going to reach one of the traps, and they're shaped as hexagons so that this allows the oil to drain out of the traps. And what you see is that in a few tens of seconds, in about 5 minutes, you can fill this complete device with hundreds of individual droplets. Each one of the hexagonal anchors contains exactly 1 droplet, and each one of the droplets contains on average about 100 cells. And again, this is in real time. And once you have the droplets located in the different positions, now you can stop the flow, and then the cells will sediment by gravity and meet each other. And now this is a time-lapse movie over, I think, 12 hours or so, or where the cells are going to start interacting together. And they create, instead of just a random aggregate, they create what we call a spheroid or an organoid, depending on the cell contents. And so after a few hours, what you see is this really nice, compact biological object that has not only a bunch of cells inside it, but the cells are actually organized depending on their phenotype, depending on what proteins they express, what stiffness they have, and so on. And you can play more fluid dynamics games by actually applying an external flow here. And you can see that this aggregate is really circular and really spherical. Okay, so I'm going to move a little bit quicker now because I think I'm running out of time. But the bottom line with this device is that on one of these devices, you have hundreds of spheroids or organoids, and you can observe each one of the cells individually in each one of these positions. So you can create, with automated microscope, you can create a really fine description of each one of these organoids, but you have hundreds of them on a single chip. And by running a few chips, then you end up with tens of thousands of organoids with hundreds of thousands of cells, and for which you can build what we call multi-scale cytometry. So cytometry means that you're measuring cells, and it's multi-scale because you know each one of the cells where it is in each one of the organoids. And so just to show you a little bit what this is useful for, if you have different cell types, you can mix them together and you can say that on average, the red cells are on the outside and the green cells are on the inside of the organoids. So here we go for the immune challenge. Now what we want to do is we want to understand the interaction between immune cells and cancer cells. And this is the future of immunotherapy. This is the future of how we're going to treat cancers. So what we're going to do is we're going to make a large droplet that contains a cancer spheroid and a small droplet that contains immune cells. And by pairing the droplets up in these microfluidic devices, we can merge the droplets together and bring the cells in contact with each other. So this is what the experiment looks like. In the large droplets, you have cancer spheroids. So these are cancer cells. And they're not just in a liquid droplet, they're in a hydrogel droplet where the cells can actually migrate. The next day after you make the spheroid, you bring the immune cells and then you merge them together. And what you get are the immune cells that are in the same droplet as the cancer droplet. And now if we make a time-lapse movie, we can follow the position of each one of the immune cells as they migrate and they explore the volume of the droplet. And then at some point, they will actually come into contact with the cancer spheroid. And this is where the biological magic happens because the immune cells recognize the cancer spheroid and they start secreting molecules that attract other immune cells. So if I look now at the late times, this is going to go a little bit quicker, but now these initial cells have recognized the cancer spheroids. And what you see is that they attach to it and they recruit other cells. And the immune cells start to kill the cancer cells. And this is how the immune system actually is able to detect and kill immunogenic cancer cells. So after about 20 hours, you've completely destroyed this. But now remember that we're working with devices with many, many spheroids in parallel. So on a single chip, we have a range of different outcomes of these cells. So the green cells, of course, are the immune cells. On the bottom right, what you see is that they're completely destroying the spheroid. On the top row, there's very few immune cells, and they're almost not having any effect on the spheroid, and there's a gradient of these things. And so I just have a couple of slides left to finish. So we're able to relate the outcome of the spheroid, is it dead or is it not dead, depending on the number of cells in the droplets. And what we're able to do is to say that below a certain threshold number of cells, they never kill the spheroid. Above a certain threshold number of cells, they always kill the spheroid. But the question is, what does the intermediate range tell us about the interactions between the immune cells? And this is a very long story, but I will just give you the punchline. The punchline is that in the intermediate range, there's a certain probability for the cells to kill the spheroid, and this probability increases like a sigmoid function. And by comparing the actual interactions between the immune cells, what you can extract from these types of data is that the immune cells don't act as individual cells, they actually collaborate together, and the collaboration is critical for actually killing the cancer spheroid. So this is a long story. It's under review in a journal, so hopefully you will be able to see it in the coming months. So to summarize, I've showed you that we started from a very basic fluid dynamics question, but then we've increased the complexity of what we do inside of these droplets until we now have multiple cell types with multiple cells collectively acting together. And this is a story that's over 10 years, where we've gone basically increasing the level of complexity as a function of time. And so I'll just finish by saying that this is work that's been, as I said, about over 10 years. So there's many PhD students and postdocs who have collaborated on this. And this is the students from the lab and the postdocs from the lab. And this has been in collaboration with several different groups a little bit over Europe. Thank you very much for your attention.
All right. Yeah, thanks very much. Do we have anyone would like to ask a live question? Charles, I'm guessing after the oil drains, can you make it so that the water droplet conforms to at least part of the solid surface to avoid optical aberration, optical, because of the index of refraction difference between?
So the droplet shape is purely determined by its volume and the shape of the anchor. So if you make the anchor small enough, then the droplet will be squeezed against the bottom, and there you don't have any optical aberration. You can play different game. Sorry?
There's maybe a trade-off. If you make it too shallow, then it's a poor anchor. But if you make it too deep, you have optical aberration.
Yeah, well, I mean, you have actually a range of parameters to play with. You can make it shallow enough that it's flat on the bottom, but it can stay sufficiently strong. But the other game that we can play is to actually remove the oil by gelling the droplets. You can actually remove the oil and replace it with an aqueous medium, and there you almost have the same optical index. So you can improve the quality like this also.
I see, to match the index. I see. Cool. Very cool.
Hello, I'd like to, I thank you for your presentation. It was really interesting. I would like to know if the temperature gradient on the very last part of your speech, you talked about the cancer spheric and how it is getting killed by the immune cells. Can the temperature gradient play a role in this phenomenon, or this procedure?
So we try to work as much as possible at 37 degrees when we have cells. So we put our devices in an incubator, even when they're on the microscope. So we almost never have a temperature gradient because the cells just don't like it when you have different temperatures. So all of these experiments, at least with the cells, they're all done at 37 degrees.